Good evening. My name is Paez. I work at Novel Research Institute in the Plant Cell Biology Lab under the supervision of Elison Blancaflor. Today, I want to show you our results on the correlation of embryonic root growth and biomass production in winter wheat. So, yeah. As an overview of my talk, I will show you first the objectives for this project. Then I will remind you very briefly the wheat root system, because we already had a few good talks about that. Then I will tell you why we did this study, how we did this study, and the knowledge that we gained from this study. At the end, I will give you some conclusions, and even if it's not very orthodox, I would like you to have the take-home message or the main conclusion of this study from the very beginning. We found that fast embryonic root growth benefits early biomass production in winter wheat. So the objectives of this project were two. The first one were to identify root traits that help improve early seedling vigor in winter wheat, and then we wanted to correlate these root traits with fall forage biomass in field experiments. Why early seedling vigor and why fall forage biomass? Because farmers in the southern Great Plains of the US, they want to use wheat as a dual-purpose crop. So they want to plant wheat at the late summer, and then feed the cattle in November, more or less, during the fall and winter, and then at the end of the season in the spring, they would like to harvest some grain production. So I am going to be focused on the production of this forage during the fall to allow to improve this forage production to feed the cattle. I will show you here the wheat root system architecture. As we have seen before, we have two main groups of roots, the nodal roots, or someone called them adventitious roots, that they grow from the very bottom of the leaves, here, marked with this white arrow, and sometimes they grow some of them, they grow from the coleoptile, but all the time they are growing from a shoot tissue, and then they are the seminal roots, these ones marked with the green arrow. These roots grow, they are the embryonic roots, they grow from the seed when the seed germinates, and they are the most important roots at the beginning of the plant growth. We already know that they are very important during the whole life of the plant, but at the very beginning, when I am realizing my study, they are important for the early seedling establishment. So I will pay attention to these seminal roots. This is our root phenotyping system, not as cool and fancy as the ones that we saw before during this session, but it works. We are using cafeteria trays. If at some point of your life you are invited to Novel, don't worry. If you go to the cafeteria, I am not using those trays. You can safely have lunch. We bought our own. They are red, bright. They are very different from the ones from the cafeteria. What I do is to put germination paper. I moisture them. I put the seeds in a line, and then I cover them with another germination paper and another tray. This is a very good method to stack a lot of trays together. We don't use too much space, and we allow the plants to grow vertical, that it makes me a very good growth to analyze the root traits that I am interested in. So when I start growing a very big variety of cultivars, wheat cultivars, we realize that there are very big difference. For example, cultivar 1 and cultivar 2, they develop longer roots comparing to cultivar 3 and cultivar 4. And in the case of cultivar 2 and cultivar 3, they develop shallower angles that, for example, cultivar 1 and cultivar 4. So I decided to be focused in these differences in root elongation. because differences in root elongation, they give me differences in the adult root system. The cultivars that grow faster, they reach deeper soil or cafeteria trays, and then they grow, they branch more. So I was focused in the root growth rate, that are the centimeters per day that the root growth, because I wanted to get rid of differences in germination. So I let the plants grow for 3 days. I marked the tip. Here I showed you this yellow dot in the primary root, the root that grows straight, following the gravity vector, and I separate the characterization of this root from the ones that they grow with a specific root angle from the vertical, that are the secondary roots, seminal roots. So then I let them grow for another 3 days and then divide the root length between 3, blah, 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 centimeters per day. This is the root growth rate. Why I did that? Why we decided to be focused on the root growth rate? Because we know there are many studies that show that seminal root traits can be related with adult root traits. And then when we do these experiments in the greenhouse or hoop houses, we can see that the roots that grow faster, they elongate more, they have more rooting depth, they branch more, they have more surface area that can explore the soil. So they give, it does give, that is beneficial for the plant, so it can grow better above ground plant part. So we will find cultivars that with better fitness in the field comparing to others that grows shorter. So my idea was to make a shortcut here and correlate directly root traits in very young plants with biomass production in the field, with performance in the field. Why? We all know that studying small roots is better. I would rather prefer to study this type of roots more than this type of roots. Mainly when we dig them out from the field, we need to wash them, we need to take picture of them. To study this type of roots is a nightmare. And also, when we manipulate them, we can break them. So it's better to study this type of roots because they are going to be less broken. And if we want to convince breeders to breed for root traits, we need to enhance, we need to speed up the process of phenotyping. So I phenotype in this way with my fancy cafeteria tray system, 300 lines. They are combining from all cultivars, elite line cultivars for grain production or forage production, and I selected 40 because I am going to go to the field and 300 lines is kind of too much. So I selected 40 wheat cultivars with differences in root growth rate in both primary root and secondary root. You can see that the primary root range from 1.5 centimeters per day to almost 5 here. And the secondary root from almost 2 centimeters per day to almost 4 centimeters per day. Okay, the phenotyping was conducted in 2 main events. The first one, the seminal root system phenotyping in the laboratory in 2016 with the plastic tray system. We studied 3 and 6 days all plants. And then we went to the field and we phenotyped during January 2017 and 2018 using a laser beam sensor platform that is attached to a tractor. And we were focusing 3 months old plants. We harvest material and we took measurements from 2 different seasons, 2016, 2017, in 2 different locations with different soil type. And then we grew 2 replicates per location. The shoot traits that we characterized were the ones that give us an idea on how healthy and how fitness of the cultivar in the field. For example, the normalized difference vegetation index, or NDVI. It gives us an idea on how live, how green, how alive is a cultivar. So for example, here, this cultivar has higher NDVI index than this one on your right. And a higher index are desired at this point. Then we phenotype the canopy height, because taller cultivars at this stage of fall forage development are preferable. So we will select this type of cultivars versus this type. And then we calculate the predicted biomass in kilograms per hectare using the previous measurements. Okay, so the results, I combine the results of both location, both season, because I want to pay attention to the correlations that occur independent of the environment. We found forage biomass production from 600 to 1,000 kilograms per hectare in the selected cultivars, in the 40 selected cultivars. And I found positive correlation between both primary and secondary seminal root growth rate and the fall forage biomass. The correlation is not extremely high, but is statistically significant. Also, for the canopy height, we found a big range of data from 4 centimeters to 8.5 centimeters in these 40 selected cultivars. And I also found a positive correlation between the seminal root growth rate, again, both primary and secondary, and the canopy height. So I show you graphically what we found. These are 2 pictures of the field plots. On the left, you find the biomass production, and on the right, you find a heat map for the canopy height. So in red color, the shorter cultivars are the ones that they produce lower amount of biomass, and in green colors, the higher biomass producer and the taller cultivars. So for example, Avalanche and New Plains are both here in these red boxes, here in for the biomass and the canopy height, and NF 97117 and NF 98109B, they are in the green boxes, both for the biomass production and for the canopy height. So the 2 cultivars that has higher biomass production and taller canopy, they grow faster, their seminal roots, as you can see here in these pictures. And they are located on the right side of the chart. While Avalanche and New Plains, the cultivars that produce lower biomass and they are shorter, they have a slow growth rate comparing to the other ones that I showed you before. And you can locate them here in this part of the chart. So as a conclusion, if we pay attention to the seminal roots of wheat cultivars, the ones that grow faster, if we select the ones that grow faster, we most likely find in the field this type of cultivar versus this type. At this point, I would like to thank everybody that helps me in this project by harvesting, planting, or washing roots, because I didn't show you the work that we did in roots, but we wash a lot of them. The forage agronomy group at Novel, they helped me with the field phenotyping, with the laser beam platform. The small grains breeding group, they helped me with the management of the field plots, and the plant cell biology lab. I would like to thank first Ellison, because he gave me the opportunity to learn a lot about crops and field experiments. And all of them because we are a cell biology lab. We don't go to the field, but they were always willing to take a walk with me and follow me in all my crazy excursions to harvest roots, leaves, and everything. So I am very thankful. And thank you, everybody, for your attention.